So let's get started. It's on. Okay, we're very fortunate today to have Professor Bob Austin of Princeton to give us the IAS Distinguished Lecture today. And Bob is well known to many of us. He's one of the pioneers in the field of biophysics. And he's been doing biophysics way before this area became a very hot area. And his research in biophysics covers a wide range, starting from the fundamentals of protein dynamics, including energy landscape, quantum mechanics of photosynthetic proteins, and all the way to DNA sequencing dynamics, and most recently he started with study the physics of cancer growth. So let me just briefly introduce Bob. He received PhD in 1976 from University of Illinois, Urbana-Champaign, and then he moved to University of Princeton, started his academic career in 1979, and he became a full professor at Princeton in 1989. He has received many award honors, and let me just mention a few. He's the fellow of American Physical Society, fellow of the American Association for the Advancement of Science, and he also a member of National Academy of Sciences in the U.S. He also is a frequent visitor of this campus. He took a sabbatical a year ago at this campus, and in fact, he's still running a lab here at USD. He's the visiting member of IAS, and we are very happy to have him today to give a talk about his recent work. I believe partially it's done here. About the title is already here announced. Let's welcome Professor Austin.
Thanks, Penker. Let's see how this goes. It's always more fun, as I mentioned, to Linus to write a talk than give a talk. So it seems good when you're writing it, but then it's miserable when you actually look at what you wrote. So let's see what happens here. So I'm going to talk about a really important subject, and I'll try to cover it in some detail, and so at the end we'll all understand something about what's involved in how bacteria evolve resistance to antibiotics, which is obviously a major subject of health concern. And of course, all this work was really done by students, pretty much. Certainly, Quincy John, who's actually a University of Science and Technology alumni. He got his master's degree here. Then for some reason, he came over to Princeton, and so I was lucky enough to have him work in my lab. And my other grad students, Guillaume Lambert and David Liao, and Christelle Robin, she's right over there. She's a postdoc. She's associated with the Institute and working in Widgie Wen's lab. And also I've worked with some theorists, Terry Waugh and Rutger Hermsen, who are at UCSD. And we actually work with biologists too, Nader Pumar and John Kim at UC Santa Cruz. So those are the guys who really did all the hard work. So here's the outline of my talk. First, I'll assume you know nothing. I'll tell you about bacteria, antibiotics, resistance. Why is the evolution of resistance a world health problem? Some statistical physics as to why this should not really be a surprise that it's happening, and then my experiments to try to go after the fundamentals of what's going on, and then a little bit about where I'm going in the future, perhaps. So let's talk about the definitions of what I'm studying here. Of course, a bacteria are single-celled organisms. They can and do act collectively to form biofilms or something like that. I've studied that, but I'm not going to talk about that today very much. They have a single circular piece of DNA, but it's a chromosome in the sense that it's compacted. Let me try to back up here. It's compacted into the cell, which is around 1 to 2 microns, millionths of a meter long. They're called prokaryotes because they don't have a nuclear membrane, however. Basically, things are free to move around to first order inside a bacterium. So they're a lot, in principle, simpler organism than a eukaryote. Like all are consisted of eukaryotic cells, which have a nuclear membrane and are much bigger. Like as I said, the bacterial DNA actually comes in sort of two forms. The main chromosome consists of a circular piece of DNA, which is continuously circular, and you usually write these genomes as a circle as a consequence of that. And I'll talk more about what I'm just showing here in a bit. And it also has these little plasmids, which I neglected to mention. I don't know where they're in this figure, actually. I don't see any plasmids. They should have put a plasmid in there. Little pieces of DNA, which and they are actually are used by the bacterium. The plasmids can either replicate themselves autonomously, or they can also join in with the B and D. They're sort of kind of view them as a little bit like a virus in that sense of the word. It's a symbiotic relationship, but the bacteria use the plasmids for all sorts of things. Okay, antibiotics are molecules that stop bacteria from growing by a couple of ways. They can destroy the bacterium physically, like penicillin. Those are called bactericidal antibiotics. Or they can prevent DNA replication. That's called bacteriostatic, such as Cipro. Or they can stop cell metabolism, for example, such as chloramphenicol. That's also bacteriostatic. So you don't actually lose bacteria here. You just prevent growth or replication, and these ones actually destroy the bacteria. Resistance is a phenomenon where the antibiotic, which used to either stop the bacteria from growing or killed it, doesn't do it anymore. And the drug industry has these kinds of agar plates where they have various kinds of bacteria. Oh, sorry, they have a bacteria which is spread uniformly as a lawn on this agar plate. Then these little things coming out here are various antibiotics. In these cases here, these antibiotics are killing the bacteria, so you see a clear ring around where this antibiotic is. The bacteria or lice are destroyed, except for here you'll see in this one right here, for instance, the bacteria actually are growing around this antibiotic. So this is an example of a bacteria which has evolved resistance to that particular antibiotic. Since the progeny inherit this resistance, it's typically a evolved phenotype. It means there's some sort of change in the gene of the bacteria that's happened as a course of getting resistance. So why is that a world health problem? Well, the first antibiotic which was found was penicillin. And there's been subsequent variants related to penicillin. They're all naturally derived. Many antibiotics can probably be found in Chinese medicine, I have no doubt. But there typically is also synthetic ones, too. When we first got this going before World War II, it was believed to herald the conquering of infectious diseases. And there was a lot of optimism. We had actually solved the problem of bacterial infections, which, of course, has been devastating mankind for eons. However, that was sadly incorrect. Bacteria develop resistance to antibiotics at an unprecedented rapid rate. Quite quickly, it turns out. In fact, many antibiotics now, such as penicillin, for example, are basically useless, and they only use it under certain circumstances. And we're actually running out of new ones. We're in an arms race. The bacteria are basically winning. And so there are only a few antibiotics which are fully functional right now. Most of them have bacteria which have evolved resistance to them, and the drug industry is running out of ideas for making new ones. So it's a big looming problem. There's a lot of literature on the subject. You can see that here if you go on the web. They call it superbugs, bacteria which have evolved resistance to most common commercial antibiotics. And you see this sort of stuff. Then the U.S. Centers for Disease Control is warning people that we have to take this very seriously. So that's the problem. So what's going on here? So it's a complicated subject, and there's some naive views about what goes on, in my opinion. Some of you biologists out here are probably going to disagree with me, but in a ways, the most common way to view it, what happens is that if you have a collection of bacteria, there are some that are resistant, that's red, and some that are sensitive, that's yellow. And under Darwinian natural selection, when you apply the antibiotic, the ones that are resistant survive, and the ones that are sensitive die. But if you keep on doing that several times, you'll end up only naturally selecting the resistant bacteria. And that certainly happens, but I think it's only a small part of the picture. Now, this is Delbert Luria statistics in which you have pre-existing mutants. I won't go into this at all today because I'm trying to speak to a general audience, but it's actually a very fascinating statistical analysis on how you would look at if you actually challenge the bacteria with an antibiotic and look at this, the way that spots appear in these agar plate cells. It's called a Delbert-Lurie analysis. Is a way to try to analyze what's happening here, and it's a fascinating thing. Actually, I have some. We're actually working on this right now, as a matter of fact, because it's actually quite interesting. And so, if you're really gifted at mathematics, you might see there's some cool stuff to be done here. I won't go into that today. We'll talk a little bit about statistical physics now, on the other hand, here, because my feeling is that in order to understand this phenomena, we have to have some understanding of it from a mathematical and physics viewpoint. And I'll try to explain a little bit about this. So this stuff here is kind of weird. I'm going to give you right now. It's sort of my own way of viewing what's happening here. First thing to notice is what physicists typically don't realize is, of course, that bacteria reproduce themselves. They grow, and they can actually grow exponentially. And I don't know anything else in the material world that actually does this. So it's quite unique. So the emergence of resistance and the principles of natural selection simply tell you that the emergence of resistance is inevitable. It will always happen, right? You can't stop it. But it's just a matter of time and space. How long does it take to happen, and where does it happen? But it will happen because the organisms follow these very simple rules. But that's not really enough to understand what's going on here. You need a little bit more than that. So what a biologist or an evolutionist usually talks about is the fitness of a species. And simply defined, the way I'm going to write it down here is, it simply says that if you have some organism which has a growth rate R0 and it has a mutation, then at the end of the day, what this mutation does is it changes the growth rate by a factor of S. So before the mutation, it was R0, and now the mutation gives it an extra advantage or disadvantage. It could be a negative number here. So that's the way I define fitness here. It just means you change the growth rate by some amount. And so what I'm going to do now is something a little bit weird, is say, okay, what happens here is that if you look at a genome of the bacteria and you change the genome by making mutations, and you ask yourself, at what rate, I'm sort of doing it inverted here because that's the way physicists do it, so it's kind of a funny way to state it. How much do you have to change the genome to get a given change in fitness? Okay? In other words, if the genome doesn't really care, if the fitness doesn't change much as you make lots of mutations, okay, then by the same way a physicist defines temperature, then I would say that's a very hot evolutionary landscape. You can make all sorts of mutations. The fitness doesn't change at all. It doesn't care. So I'm going to define temperature here in Darwin's, not in Kelvin. And if a genome is a weak function of the fitness, then it's a very hot environment. On the other hand, if you change the genome and the fitness changes very dramatically, in other words, that means you're in a very selective environment which really cares about the sequence of the organism. And if you change that, then your growth rate goes to 0 and you just die out very quickly with any small changes in the genome. Then I would call that a very cold Darwinian landscape. If t equals 0, of course, and this is infinity, you can't change the genome at all because the fitness change is huge when you try to make a few mutations. So at t equals 0 Darwin's, there is no evolution. Okay. So, I mean, for the physicists out in the audience, I'm sort of defining temperature in Darwin's the same way a physicist defines temperature in terms of the entropy, the disorder in a system, and the internal energy. The change in the entropy with internal energy of a system is 1 over the temperature. Same way here, that the entropy is simply the genomic space. The both, in this case, are a matter of disorder. And the energy is the same as the fitness. So this temperature in Kelvin and temperature in Darwin's are related to one another. And, of course, Boltzmann, who died, committed suicide because nobody bought into the concept of atoms 100 years ago. He defined the entropy of a system as the log of the number of possible combinations, number of states the system had. It's on its tombstone. And the same way here, I can define the genome, in this sense of the word, as the log of the number of changes you can make in a genome and keep the fitness the same. So the more changes you can make, the more disorder there is in that genome, and the bigger the genome is, just like the entropy is related. So they're connected to one another. And by the way, I've also introduced a whole new number here. Aren't you lucky today? This, instead of Boltzmann's constant, this is Darwin's constant, which connects the disorder in the genome and the genome as the way I've defined it from this temperature. And so that means that, unfortunately, I don't know the value of Darwin's constant, so don't ask me what it is. Okay? So just like in the log of when you look at states in a system, if you have a ground state and an excited state, the log of the number of states for a given temperature that are created that is the measure of the internal energy of the system here. That's from classical statistical mechanics. In the same way, I can define now the fitness. The bigger the number of individuals you can have at a given Darwinian temperature is a measure of the fitness. And then what you would do now is actually define Helmholtz free energy. And the reason you do that is that in physics, in statistical physics, the free energy is the thing that's minimized in a system. It's a function of both the internal energy and the amount of disorder. And if you don't include the entropy in a system, you can't begin to understand why the system goes to that minimum that it does. So that's why physicists came up with the Helmholtz free energy. So I would do the same thing here. I would define a fitness, or free energy for our evolution systems, which is a function of both the fitness that it has and the disorder in the genome that's possible to give that fitness. And again, I'm going to define it to be the Sewell-Wright free energy. In a way, what will happen in a biological ecology, it'll try to minimize this free energy. That's what it'll do. And it's a combination of growth, which is this, and statistics, which is this. Just like an apple will fall off a tree and hit Newton on the head because it's minimizing the free energy, so a system will go to a minimum in this free energy space. The only question is, how fast does this happen? It could happen very slowly and wander around. If the system is at some place here and wants to go over here, it may be very inefficient wandering around to get there. Or there might be ways to very rapidly move to the most fit place. And the bacteria seem to be able to do this and not this. They've learned how to very rapidly move from local minima to the biggest minima in this free energy. That's the question, is how fast does this happen? Okay, so you can sort of view what happens here is that I sort of defined a temperature, and what happens is that when you put the system under stress, you can sort of view the evolution as a flow of this genome space to a region of the minimum free energy. And I'll give you another, there's a reasons why I'm boring you with all this crap. You could ask yourself, well, when the system flows to this new minimum in the genome space, is it a unique new species, or is it just some mess, right? And so that's sort of like in physics asking, when you take water and you cool it down, does it just turn into a glass, or does it turn into a crystalline solid? And that's the difference between phase transitions. And you can do the same thing here in evolution dynamics, by the way. You can ask yourself, well, there's just a big splash of different sequences that happens when I give some antibiotics? Or does the thing go to a new species when it happens? And that's the same as a thermodynamic phase transition. And obviously, we could not have talked about phase transitions if I didn't tell you how you compute a free energy. And I made one for you, so you're welcome. I did that. And I won't go into the details again, but the way the physicist works with that, he simply takes this free energy and he expands it out as a terms of genome and temperature. And that's this called the Landau I've lost my words here. Anyways, you expand it out in a Taylor series kind of thing. And you put this negative sign here, it's very important. But it's an interesting thing that happens, actually. If you take a system in a very hot place where it doesn't really care what the genome is, then it'll have some sort of average sequence. But if you cool it down by giving an increasingly greater stress, if you apply antibiotics to it, in this expansion I did of the free energy, what will happen here is that it'll suddenly jump from one sequence to another sequence, which now becomes the new unique minimum. So it's possible in evolution, if you write a free energy and do this experimental, writing down as a free energy, that the system can actually make a first-order phase transition from one species to another species as you apply stress to the system. And you can make a fairly exact analogy between thermodynamics and evolution dynamics in the way I've done it. Okay, so that's all very boring, but I wanted to say that there are ways you can write down formalism between the physics you've learned, hopefully, those you've taken physics as undergrads, and what happens in a biological system, that there's some fairly general principles one can write down. But of course, as my student from UST has pointed out, everything's garbage. He uses stronger words without experiments. So these kinds of theoretical things are sort of useless until you actually do experiments. So let's talk about now in experiments which try to see whether or not this is what's happening in the evolution of antibiotic resistance. So what I do is, as Penger mentioned, I use microfabrication technologies to try to create these kinds of landscapes of free energy and find out how fast the system moves to it. And because I need a landscape, because it has to flow over this landscape, I don't use flasks because that's just one environment. It's like a physicist studying one ideal gas and never going to something more complicated. So I don't do this kind of conventional microbiology that the microbiologist does. They tend to get upset about this because they sort of feel that if it doesn't occur in their flask, in their test tubes, that it's not relevant. But my point is, I just came back from Beijing. I got sick there in Beijing. I got a bacterial infection, I think. It's not as clean as Hong Kong. It's dirty. It's very complicated place. And the way evolution proceeds on the streets of Beijing with the bacteria is a lot different than what happens in a microbiology laboratory. And so that's also true about a lot of other places too. So I'm just telling you that you might see different emergent phenomena in a more complex space than in a simple space. And they may really be quite different. So actually, as Penger mentioned, actually when I was on sabbatical here at IST at the Institute for Advanced Study, I designed and built actually in your nanofabrication facility with Chi Kwan Tung. I should have mentioned him here. I think maybe, anyways, he was a postdoc I sort of shanghaied over to Hong Kong for 1 year to help me out when I was here. I came up with the idea for this thing called the Death Galaxy. So it really originated here at the Institute for Advanced Study in UST. It was made here in Hong Kong. So before I had come here to Hong Kong, I had been using micro technology and nanotechnology to work on ecologies, because I knew that ecologies were very important for studying biology. And I tried to make, so an ecology has to be rich. It can't be simple to be really biologically relevant. So I made these microfabricated things and had bacteria growing back and forth. And you have nanoslits here that bring in food. But it was a one-dimensional thing. So I was sort of stuck in the physics of one dimension, right? And then I finally made the giant leap to 2D here in Hong Kong to move on to more interesting phenomena. So where am I at here? Is that right? So the question I came up with when I was here is, if you apply a very large temperature gradient in terms of the way I've defined temperature here in Darwin's, using antibiotics on a complex two-dimensional landscape, could I greatly enhance the rate of evolution over what's typically seen in a microbiology laboratory? And by the evolution means the flow of genomes over this space. So could I see rapid emergence of antibiotic? And could I actually see whether or not it was a phase transition kind of phenomenon actually occurring there? So what I do here is a very simple idea, actually. I have food flowing on this top half here, and food plus antibiotic flowing in this bottom half here on the outside perimeter of this 1,500 microhabitats. And I put in here an antibiotic here and none here, so that there's a huge temperature gradient in Darwin's that exists between here, which is very hot, and here, which is very cold. The bacteria really can't grow here, and they can grow here, but they'd like to move over here because there's a lot of Lebensraum, a lot of place to live down here, but unfortunately this is antibiotic here. So the idea here is by making a very complex gradient that I would actually find that the bacteria can very rapidly evolve resistance to antibiotics, just like on the streets of Beijing, apparently. So that's what the thing looks like. It's about 2 centimeters across. And if you just pump in some dye around the bottom here, that's more or less the way the antibiotic concentration varies as a function of position. It's kind of funny looking because it's a mixture of diffusion. The nanoslits right here, you can sort of see them, are only 100 nanometers deep. I don't allow the bacteria to get in and out. They're stuck in here. But there's also flow. There's pumps running. So it's a mixture of diffusion and advection here. And the idea is what's going to happen now is if a mutant occurs, he's able to now to move in this landscape over to some other place where the free energy is even deeper. And it can quickly freeze into a new genome. I could view this in I have different ways of mathematically representing this. Today I'm giving this in a thermodynamic view. So this array of ecologies is not isothermal. And we're going to look at the movement, the heat transfer, if you want to, of the genome from one place to another place. So that's what the device looks like. And I had to pick my antibiotic I wanted to work with. So I chose Cipro. Cipro is a very interesting antibiotic. It's commercial. I've taken it myself when I had an infection once. And it looks like I'm actually, whatever infected me was actually resistant to Cipro. It didn't help. I had to go to another antibiotic. But it's a very common one. It's a very potent antibiotic. It's one of the few ones that work. The reason why the doctors like Cipro is that things like penicillin and stuff have a lot of side effects because they attack a lot of things. Cipro is actually quite specific. This is an artist's conception. You'll see a better picture later. It binds to something called gyrase. Gyrase is a protein which takes the double-stranded DNA, and when you try to replicate it, I won't go into the topology of that, but it's a knot problem, basically. It's a topological problem. You have to take out what's called supercoils in the DNA in order for the replication to continue to go along that circular DNA, because it is a circular chromosome. So that's the gyrase's job, is to cut the DNA, let it unwind, and then relink it back up again. That lets things go on. And Cipro binds to gyrase and blocks it. And it's quite specific for the gyrase in bacteria, and it doesn't affect you very much at all. You can take a lot of Cipro and not get sick. That's why they like it. The trouble is that when that happens, when the Cipro blocks the gyrase, the system notices this. It says, what's going on in sector A? What happens is because you can't reform that cut DNA that happens when it tries to replicate itself, you start making single-stranded DNA. And the system knows that single-stranded DNA is a bad thing. It should always be double-stranded. Why am I making single-stranded DNA? And a lot of physicists study this. Then there's something called REC-A. It activates a whole network, something called LEX-A. And the interesting thing that happens is that the LEX-A ends up making new error-prone DNA polymerases. In other words, the new polymerases that get made when this signal gets set off actually makes mutations in the copies of the DNA that it makes. And the idea is that these error-prone polymerases eventually will come up with an altered gyrase which doesn't bind Cipro. Now the organism has mutated. It's now resistant to Cipro. So it's what's called a genotoxic because it binds to the gyrase and screws up DNA replication. It's bacteriostatic because it doesn't kill the bacteria. It just makes them join the Catholic Church as a priest or a nun, and they can't replicate themselves. That's Cipro. I won't go into the network stuff. I just came back from a meeting in Beijing where we analyze, I don't do this kinds of stuff, but lots of physicists and mathematicians like to analyze these kinds of networks. It's interesting here is that what the system does is normally the error rate for DNA replication in a bacterium is quite low. Only one base pair in 10 to the 9th per replication is typically changed. That's the error rate at 10 to the minus 9 per base pair per replication. And the system works pretty hard to make error-free DNA synthesis. If something goes wrong, stalled forks, single-stranded DNA, it works at it when the DNA damage. However, if things get up to be too much error, too much single-stranded DNA is building up, then this Lex A thing triggers it off into a new state. That's this other state here. This is called the SOS response, save our souls. And when the SOS response is triggered, then the error rate for making mutations goes up by at least a factor of 100. So the system knows when this happens. It tries to do the best it can, but then it makes a phase transition over to another state. If you're a physicist, there's lots of fun here. If you're an electrical engineer, there's lots of work here to be done in analyzing this kind of network dynamics. We're not going to go over that. However, the idea here is, by the way, that these gradients, these thermal in Darwin's, is sharpest and steepest where the gradient is highest, just like in thermal conductivity. If you want a lot of heat flow, you want to have a high thermal gradient, fixed law. The same thing happens in my device here in principle. So although I've got hot and cold, at these apex corners here, I call them Goldilocks points. If you know the German fairy stories about Goldilocks and the three bears, Goldilocks always finding the best thing. These are where we expect to see evolution to ignite. At these places here where the gradients in the Darwinian temperature are steepest, we should see evolution ignite here or here in my kind of weird world. So when I do these experiments, here's what we expected to see happen. We expected to see, I was making a free energy gradient in this device, a complex landscape. Expect to see emergence of a mutation in the regions in the presence of stress in a hot region where mutations are cheap and easy, and then flow of that mutation into a colder region. And then once it goes into that region there, it would freeze in some sort of a phase transition to form a new strain of bacteria, which is now resistant to the antibiotic, which created this genomic temperature gradient. That should be the process. So here's the experiment. Put in 1 million bacteria. This is running here in minutes. So this is the inoculation of 1 million wild-type bacteria. Remember, the gradients are there. We expect to see evolution to occur at these 2 points right here. Hot here, low Cipro, cold here, high Cipro. And after about 3 hours, you suddenly see the emergence of resistance to Cipro in this region here of very high gradients along the ideas I was just trying to propagate here of a thermal statistical physics for evolution dynamics. And then what happens now is that once the strain of bacteria emerges with a genome which now has fitness to the antibiotic, then it'll now spread throughout the entire device. And by about 10 hours, you'll see the bacteria now growing everywhere in this region. And by the way, the Cipro here is extraordinarily high. It's about 200 times the normal dose a doctor would give you in order to, if you have an infection. So these bacteria here are bad news. They will grow in extraordinarily high levels of Cipro. And if you ever got infected with this bacteria, Cipro would be completely useless to you. But the interesting thing here is that the claim is that this actually happened in 3 hours, not 30 days or something like that, because of the way I designed my device. And you can see it, maybe see a little bit better here in this histogram. You put in 1 million bacteria, and they will spread out. They get at the food at the edges. There they're going spreading out. That's a process called chemotaxis. Every one of these subjects you can spend your life studying, by the way. And then right here, in this region right here, this Goldilocks point, we have ignition of resistance to Cipro in about 3 hours. And then these resistant mutants then proceed to flow along this fitness landscape this free energy kind of thing, out along the periphery of the device, and now you start to have bacteria growing throughout the chip at very high levels of Cipro. I was amazed when I saw this happen. I didn't think the whole thing would work, by the way. But it does, apparently, if you believe it. So, well, okay, that's pretty neat. But wait a minute. If you go to Wikipedia, everybody always talks about there is pre-existing mutants. And then you do natural selection. You have a final population. I understand that. But I said that's not what's happening. Here, actually, I started out with the bacteria which were not resistant to Cipro, and they evolved in 3 hours de novo resistance to Cipro. And this is a heretic view for most microbiologists. They view that this is the way it happens. It's always pre-existing. It's like turtles all the way down for the origin of the earth, right? It's something happened way back when, but not now, right? So, but there's a test for this. It's very clear. Okay, well, if there were pre-existing guys, and there must be very few of them, by the way, then if I studied in 1 million bacteria, if I put 100 bacteria in, then surely there'll be no emergence of resistance, because there'll be no pre-existing mutants if I only have 100 bacteria. There might have been 1 in 1 million that did it here, but certainly not with 100. So we can do that. See if this works here. I'm not sure I can start this movie with this thing here. Let's try this. Yeah, okay. So this is an experiment where I put in 100 bacteria. And you can't see them with my camera. They actually were there, but there's nothing visible. Now remember, 3 hours before, we already saw the ignition of resistance to Cipro in 3 hours. And so, well, things are not looking great here. Maybe Lehrbuch and Dury were right, and I've been wasting my time. That all resistance starts from pre-existing mutants, and this junk about things de novo evolving rapid resistance can't possibly be right. The numbers can't possibly work. Now we're up to 13 hours. The advantage, of course, is this experiment just simply runs. The grad student can simply go home and watch TV or something like that, or take the weekend off, right? And you can be very patient here with this kind of experiment. You don't have to be constantly changing plates or something like that. Still looking bad at 20 hours, actually. Now we're getting on to a full day after I've started this experiment out. But now you see something is starting to happen here, right? You see this flickering is actually the emergence of bacteria growing in this chip. And by 28 hours, you now see the bacteria actually growing everywhere in this chip. And they actually form these beautiful rings, and it's fantastic, actually. Quite a bit more interesting pattern than the high inoculation rate. And I suspect these bacteria have evolved along quite a different path than 1 to 10 to the 6. But the fact that we saw the emergence of resistance with 100 bacteria, I would say is certainly a claim that this is de novo, that they figured out something from the start. There was no original guys there which already had their resistance intact. And you can do a series of experiments. You can start out with 1 million, which evolves resistance very rapidly, and go down by factors of 10, 100,000, 10,000, 1,000, 100. And you inevitably see emergence of resistance. It simply takes a longer time for it to emerge when you have a lower. So it's quite clear to my mind that this is true de novo evolution of resistance by the bacteria. It's not pre-existing. When you want to argue with me about it, we can do it later. And you can actually write down some mathematics here to try to understand this a little bit. You can talk about the way that the wild type grow. I won't go into all this stuff here. This is a logistic term here, by the way, which means they compete with one another and they can't grow to arbitrarily high concentrations. This is called the carrying capacity. And they jump back and forth between these different environments. And the mutants arise from the wild type. So there's this mutation rate I talked about, mu, and they did the same kind of logistic growth, but with a different growth rate, and they also move back and forth. Once again, if you're a physicist, you can have a lot of fun. This is actually called the Fisher-Kolmogorov equations, by the way. They have a name, and they actually have, believe it or not, they're nonlinear, of course, equations. They look horrible, but they actually have wave-like solutions. One of the predictions is, which Fisher and Kolmogorov brought out in the 30s, actually, that you could actually have evolution proceeding as waves propagating over a landscape. And so you can play with these equations if you want, and I could do that, but again, life is short, so I'm not going to do that. We just did some mean field, as I say, solutions of the equation. And we simply say here that when you see, as you drop down the number of bacteria, that it takes longer and longer for the emergence of resistance. The data here is on the left-hand side, and we run these equations on a computer and put in some numbers for what we think are the mutation rates and the growth rates. You'll see that the basic pattern that the bacteria have can be reproduced by these equations for this de novo. So the mathematics, which is interesting, but it turns out, does in general reproduce the basic form that you see for the emergence, and we understand the delay that happens as you go to lower and lower. It should make sense to you that that's what happens. So I'm quite confident that this is de novo rapid evolution occurring in this device. I'm not going to pursue the waves. Again, every one of these things I could spend a lifetime on doing. All my point here is that evolution is de novo and fast, ours. And I would say it's fast because the landscapes we made, if you do these experiments, and I've always been challenged by biologists, and they think they can do it with test tubes and agar plates. And I won't go through all the tests we've done to answer that because I take it very seriously. It never works. So you really need to buy my chips if you want to see this happen. Okay? I'm not selling them. I give them to you, obviously. But every test we have made confirms the need for a stress landscape that is not isothermal for rapid evolution. That's the way you get it. And what about this the first-order phase transition crap I was talking about? It's sort of talking about that as a system moves into a cold place, that it sort of becomes bistable, then locks itself and freezes itself into a new genome. That's sort of the prediction that comes out of this kinds of stuff, too. It's not really prediction. It's an analogy, but it might happen. Well, the only way to answer that is by actually doing sequencing. So in sequencing, you find out what exactly happened in genome space in that cold, free energy landscape. Now, I have to warn you that it's actually hard to do sequencing. It's a lot of work, and I've gotten pretty serious about it. And right now, at this point, I cannot do spatial or time-dependent sequencing on single bacteria to tell you what exactly is the path that what you see happens. I don't know that yet. We'll find it out. But I'm not at that point. So once again, I'm sort of doing an ensemble average of what happens. We do replica plates and grow it up, and then we go ahead and do the sequencing. And I've learned a lot about this, by the way. What they do, by the way, in sequencing, if you don't know what's happening in sequencing, it's all the rage. What exactly happens? You've got to be careful. You know, there's a lot to this, some games they play. They take Pinger's genome, and they break it up into little 200 pieces, 200 base pair pieces or so, maybe 100, depends on the read lengths, as they call. And they sequence just that little piece. But they don't know where in Pinger's genome that came from. All I have is a solution with 200 base pairs, pieces, millions and millions of them. They can sequence those things. And then the question is, well, where the hell does that go? So they assemble that with a computer. Right? And so when they do that, they take these pieces, and then they line up these pieces using computer algorithms and create what they think is the correct combination using some very sophisticated algorithms. And you don't do that just once, actually. You have millions and millions of pieces. So you can make many of these genomes. All of them should be identical to one another, right, if the computer is running right. And so that's the coverage. So I'm showing here the coverage. This is one genome. Here's another genome. I only show 10 here. That would be a times 10 coverage. But we typically are running times 100. So the computer builds up 100 genomes that it thinks represent a particular genome. And we compare that to the control, which is the bacteria before it entered my chip. And so, but the machines make mistakes, of course. So you'll see here, here's a mistake here. We sort of knew what the sequence of this E. coli K2F should be, but in this particular genome here that it made up, instead of having an A here, there's a T. And here's a G here, right? So these are mistakes, but you'll see that they only occur in that one thing that built up. So you wouldn't call that real. You would say that's noise. The machine screwed up, or the computer algorithm didn't work right. So that would be your control will have noise in it. And these are from our experiments. And you'll see here that when our SNPs, as they're called, single nucleotide polymorphisms, you'll see that these C's here, it should be a T, it sees a C, and it goes down 100 sequences. Every single one of the sequences the machine made up has this T to C replacement. And that's times 100 coverage. So that's how you decide whether or not something is significant or not, but it's a call on the machine's part. So while you can get your genome sequenced for $10,000 in principle, it'll be garbage. It'll have so many bad calls and stuff, it's basically useless. To really do this right requires a lot of money and a lot of time, even with E. coli. I learned that lesson the hard way. So this is a sequencing noise curve. In other words, you can see one base pair change out of my... There's a lot of possible SNPs, and you'll always, when I make these things, you'll always find 100% of the time there'll be one base pair wrong somewhere in these 100 ones. And as you get as the percentage of that mutant gets higher and higher, it's less likely to happen. And you'll find out that the true SNPs, which are 100% of the time in all 100 of the sequences there, they stand out like a sore thumb from this noise. So those are the SNPs that we call. We're quite conservative when we see our SNPs. So you've got to be careful here. You can come up with a lot of bad conclusions. So when we do it, here's the circular genome of E. coli. We get 4 SNPs. All right, just 4. There they are. There's two of them here in a local region, close together, one here and one here. Just 4, not more. And the another interesting thing here is these various lines you see are different experiments. So we repeat the experiment. We've done it 10 times and sequenced in depth at a times 100 coverage every time. And when we do that, we always see only these 4 sequences. So the system comes up in a few hours with a frozen genome, which is highly specific. It doesn't come up with just some garbagey thing, which by hook or by crook works. It actually only has 4 SNPs, and it does it really fast, and you have over 4.6 million base pairs. It seems to have found 4 SNPs which do the job to give extremely high resistance to Cipro, which is really, to my mind, quite remarkable. And you can actually, the great thing about E. coli is you really understand this, by the way. Like this one thing here, this SNP here at position 2,337,183. That's 7 significant figures. That's a single SNP. We know where that is. It's been annotated. In other words, if you tell me where there's a SNP in E. coli, I say, oh, well, that's interesting. That's gyrase A. That's the damn thing that Cipro binds to, right? So it's gone ahead and made a mutation on the gene which codes for gyrase, and gyrase is the thing that Cipro binds to. That all makes complete sense. And you see, in fact, that it did a single, what's called a missense mutation. That means it changes one amino acid into another one. It changed aspartic acid into glycine, which means you lost this big group here and just replaced it with a hydrogen. That's what it did. And then you go ahead and look, we even have a structure of gyrase A. It's a miracle, right? There's a structure of gyrase A. The green there is the DNA. The Cipro is known to bind right here. That's what it really looks like. And lo and behold, the mutation that comes up is right across from the Cipro. It's fantastic. Right in 3 hours, it found a mutation right across from the Cipro, and it makes a bulky group less bulky. We don't know what really happens, but it probably allows the DNA to spin inside the gyrase and let it work. It's probably true that Cipro simply blocks the DNA from spinning, and it gives some room here for the DNA to now turn. But it actually makes complete functional sense what happened. That's the one of the 4 we found. And every time it comes up with the same one. But usually one SNP does not give resistance to the bacteria. This is a small reading. I'm sorry about this. The only point in this thing here is that usually it takes multiple SNPs for resistance to antibiotics to happen. So we know we needed gyrase A. For sure we needed that. And we hoped we would see a mutation there, and that's exactly what we found. It was a great feeling, by the way, to see the biology actually makes sense. For a physicist, it's remarkable. But we found another one. There's another missense. Missense, again, changes one amino acid to another one. It was at this location right here, 3933247. Remember that number. It's important. Again, it's 100-fold down. It's really deep. And this is an interesting one. This is a pump. So bacteria can do, there's a specific mutation here which acts directly where gyrase binds Cipro. And then these pumps are used to get rid of poisons that the organism has identified as bad news. So this is what's called a ribose ABC transporter complex, and it's known to give rise to resistance to antibiotics by pumping those things out. And so apparently it's changed the pump. We don't really know the action behind it with a single snip, which is making the thing presumably pump Cipro out more effectively than it was beforehand. And then we also found 2 more SNPs. And those were an interesting region. It turns out there's something called the MAR operon. It's something that gives rise to general resistance to antibiotics. So it's a war out there. You think your life is tough. The bacteria, it's a much tougher life. And so there is a regulon, as it's called, which gives rise to multi-drug resistance. These are the superbugs. And so it's gone ahead and done 2 mutations in the superbug place to give rise to even further resistance. So all these things seem to have gone on very rapidly in very purposeful ways. So we find out, we didn't scratch our head, gee, why the hell is that SNP there? That makes no sense at all. When we actually looked at the SNPs, oh, I get it. Yeah, it's physics. I understand it, right? It makes sense. So that's what we've done. And every time we do the experiment, so the thing seems to have evolved, a highly evolved, new, stable genotype, which is very functional and very lean. And it did that really rapidly. Okay, so that's a surprising result, and we're getting this published in a high-impact journal because it's a pretty cool result. And I'll try to tell you a little bit about where I see the future going. So what I've done so far is developed a free energy picture of evolution dynamics, and I use this physics model to devise a free energy landscape, and then I built the free energy landscape to fast-forward evolution, and the whole damn thing worked. That's remarkable. Of course, I lied to you so far. In biology, it's always more complicated than you think. So don't think you can walk out of the door here and say, oh, I understand now how the evolution of antibiotic resistance works, thanks to Professor Austin, because you would be misled in that case. So I'll try to be honest to you. I've tried to give this a hydrogen atom way of viewing rapid evolution, but it's inevitably more complicated than you think. The organism does other things too. So obviously, this thing has done something very creative, right? Hong Kong is a creative place, and these bacteria are also creative. They went ahead and changed the gene, right? But that requires a lot of imaginative work by the bacteria. A simpler thing to do is just make more copies of it. If you're having gyrase being blocked, then just make more gyrases. Maybe it'll compensate for that. And so in many kinds of evolution of resistance, you see the system takes a shortcut. It simply increases the number of genes involved in the blockage. It doesn't try to change them. It just makes more copies of them. So Sarub is working with me in Princeton Physics. We did another experiment. He thought, Bob, you're full of crap. Maybe these SNPs are there. I don't buy this. I think what happened is it simply made more copies of the gyrase gene. That's what it did. That's how it just simply made more copies of these blocks. And if you understood the sequencing technology, because it breaks up into little pieces, if there's extra pieces, it simply throws them away. The computer simply finds the pieces and tries to patch them together. So you have multiple copies of a gene. The sequencing machines don't know. They don't know. They can't tell that very easily apart. So what we went to the Affymetrix gene chip, which has most of the 4,377 E. coli genes. And that chip has actually little pieces of DNA. And it sees how many pieces of the DNA are there. It doesn't sequence, but it looks to see how many copies of a gene that are known to exist there. Okay? So we did that. So he was convinced. So everybody attacks me all the time. He was convinced it was making multiple copies, and I was full of crap as usual, which is true. But anyways, so we did that. So this is a little hard to see. I'm sorry about that. But the inner ring here, there's 4,000 genes here, right? So we try to annotate them. These are various names for the genes. This little ring inside here is the actual copy numbers that we saw. So if it's blue here, it made the same number of copies as the wild type. So when you say blue here, it means nothing changed. But you see there actually is a hot spot. It is also true that the bacteria made more copies of certain genes. as well as mutating these genes here. And by the way, they did not make more copies of the gyrase, they simply mutated it. But there were extra copies by a factor of 3 in this region right here. And I told you to keep an eye on that number because it turns out that where we got our mutation before, which is this red line right here, is what's called the origin of replication. So when the DNA is replicated in E. coli, it starts right here and starts to make copies. And if you were like a business guy or something like that, you might think, well, if something's going to get screwed up in my DNA replication, the one thing I really need is energy, because it takes energy to run everything in life. So it turns out that the ATP generation genes, the ones that code for making the currency of energy in a cell, are right near the origin of replication. And that's where we saw multiple copies happen. So apparently the multiple copies were in fact made and they look like they were the ATP synthetases. So in other words, the system put in more power plants. That's what it did. But it didn't change the number over there. That was interesting. So they've not evolved more copies of the genes in the gyrase A, which is over in this area here, but they did decide they needed to produce more energy, and they did that. Another thing I've been lying to you about is I'm sort of viewing bacteria as single organisms. They are communal. They're not solitary. They share DNA. They share signals. And if you don't understand that, you're going to never understand bacteria. So in bacterial resistance, what's happening here is both the single cell stuff I've been telling you about, but also they are communicating with armies. You're really dealing with an army of soldiers. It can be viewed as acting for the communal good. They are communists, not capitalists, right? Which would, of course, explain their success, as we all know, right? So the sharing of DNA is called horizontal gene transfer. And we now know it as a major component of evolution. You can sort of quote Karl Marx here. From each according to his ability to each according to his need. If somebody comes up with a solution to the problem, it starts generating little pieces of DNA which float on the solution. The other ones pick it up and incorporate it into their DNA. It's remarkable. And so they're capable of doing that as well. And we now think that actually it's it actually screws everything up. It even makes the whole concept of a species for bacteria questionable because they're constantly sharing their DNA. So I've tried that experiment. We're working on that now. I've ignored that aspect. I have not tried to, in a serious physics way, understand the dynamics of what we've actually seen. We're not at that point yet. But one thing I've got to get clear is, are they sharing the secrets or not? Is each one evolving separately, the answer? Or is some guy getting it and then squirting out the solution to other people? And so the way we're doing that experiment is we have a chip with two interconnected microhabitat patches, as they're called. On this side, we put in a bacterium along here, which glows red. And that red comes from a plasmid, which also gives resistance to an antibiotic here, the red antibiotic. And on this side here, we have bacteria which glow green, and they have resistance to this antibiotic here. So they are one green on here. But the question is, if there's evolution occurring in this temperature gradient, is it possible for the red bacterium, for instance, to find a solution to the other antibiotic and then squirt that over here and then turn this guy on here, and it'll carry over with it the other color? So we'll start seeing red guys appear here and green guys appear here if they're sharing their bacteria, their solutions to the antibiotic problem that we give it. This is an experiment I'm just starting to do, and it doesn't come out that well. So we're just starting to figure out how to do this. I just finished this experiment, started with a senior thesis at Princeton. And it's probably hard for you to see, but what we're beginning to see now is that we're beginning to see the green guys appearing in the red place and the red guys appearing in the green place. Just starting to do that. I simply have to tune this experiment up to see this, but I'm quite sure eventually we'll see a true socialism develop here as these bacteria start to share their secrets. I'm sure that's happening as well in this system. So I've jumped over many details here. There's a rich landscape of theory and experiment at many levels. You can spend your lifetime studying any one of these things I've talked about. However, the point I want to make is that there is no cure for the rapid emergence of bacterial resistance. It inevitably happens. And the way you can deal with it is understanding the rules by which it happens, and then trying to minimize the rate at which it happens. It's an arms race. We're always behind in it, by definition. It's the Red Queen effect from Lewis Carroll. It takes all the running you can do to keep in the same place, and that's the situation we're in now. We realize that now. That's the battle that we're faced with. And I hope I outlined for you a few of the reasons how this happens and why it happens, and then you begin to come up with ways to keep the arms race under control. I have bigger fish to fry, of course. I think the dynamics of evolution in cancer and the emergence of drug resistance in cancer chemotherapy follows the same rules I have outlined here for bacteria. So we've published an article about this. There is a connection between bacterial emergence of antibiotic resistance and the emergence of resistance in cancer to chemotherapy, which is why we've been unsuccessful in curing cancer over the last 50 years, just as we've been unsuccessful in curing infectious diseases over the last 50 years. And some infectious diseases are coming back because we're incredibly stupid about it. We've not understood these basic rules. So I think that a realization that you should get a deeper understanding of the physics and mathematics of evolution and will lead eventually to some sort of a peaceful coexistence with these phenomena once we understand them at a deeper level than we do at present. So with that, bacteria of all lands unite, and thank you very much for your time. Appreciate that.
So as usual, Professor Austin's talk is intriguing and with very deep thought. So now we have open sessions for questions.
Yep.
Did you have mentioned, but I didn't catch it. In the last experiment, the
Yeah.
red one and the green one,
Yeah.
how do they communicate with each other? How do they communicate?
Oh, okay. He asked, what were you doing in the experiment with the red and green ones? So there's actually nanochannels that connect the two. So the bacteria can't cross, but the DNA can. So we create a barrier which is diffusional in nature. So if we see this lighting up occurring, it must have been by the transfer of DNA material across these nanochannel barriers. I'm sorry I didn't make that clear. Yeah, they're connected diffusionally.
But bacteria cannot just swim to...
They can't. No, they can't. They can't do that.
Okay,
Right.
thank you.
If you can check that out, you can. Bacteria can deform themselves quite facile, but they can't do 100 nanometers. So yeah, they can't cross. That's a good question. But even if they did, they'd still die over there anyways. So if you see something growing there with the wrong color, it's not that he swam over there. He's evolved resistance. But he must have got it from his brothers on the other side of the ocean. Yes, sir.
So you study the bacteria in your device, right? And
Right.
a function of space and time.
Right.
So can you estimate your KD?
Great. So he wants me to know what's Boltzmann's. What's Darwin's constant? I've not tried to figure that out yet. I just came up with this idea, this way of viewing it a few weeks ago. But yeah, the trouble is that I have to know how if you look at the way I define it, I have to know how the fitness changes with the genome, right? That's the connection, right? If you look at what's going on here is that the Darwin's constant connects how much disorder there is in a genome for a given fitness, right, with that concept of G. And so that means I have to figure that out. So unlike Boltzmann's constant, I think, which is a universal constant true everywhere, I think my constant is not a constant, right? It's going to be a function of the environment. That makes some sense? So it's sort of a misnomer to call it a constant. I could define it for a particular region, but it's probably this whole approach of using thermodynamics and stat physics in this conventional way is probably a good analogy. Whether it's a computational tool, I'm not sure at this point. I'm not sure. I don't know the number. Yeah.
Disorder in the genome certainly involve both genes and everything else.
Right.
So you just mainly dealt with genes so far. Isn't it?
Say again.
So disorder in the genome obviously concern the whole genome.
Oh, yeah. What do you mean? Yeah, the disorder simply means if you change the genome, how does the fitness change, right?
So you mentioned mainly...
But it's everything, right? In other words, the genome ends up with a phenotype, and that gives rise to a fitness, right? So it's a long filter between... That's why I can't calculate it, right? I mean, you can make mutations in E. coli and ask yourself, does the growth rate change in a particular environment, right? That's the disorder in that genome. If you follow what I'm saying there.
Not the physical sense.
No. Well, what do you mean?
For instance, maybe for a biologist, I may be more interested in the structural part of the genome rather than the coding part of the genome. And
the structural part of the genome. What is it? What do you mean by this?
The non-coding part.
Oh, then, well, in E. coli, everything's coding. I do believe. Is that right? I think it's a very efficient genome, right? So if you want to go to eukaryotes, we're only like what few percent of it is actually coding, right? Then that's a much more, but it's still, the disorder means any changes anywhere in the genome, whether it's coding for a gene or doing structure.
The price of the cost of sequencing going down, then your system is able to address the difficult things for a biologist,
right?
The non-coding part,
right?
And your entropy and your disorderness may have something to
right. Well, yeah. So the idea, of course, we're going to be moving over to eukaryotes. We're actually doing eukaryotes right now. So to what extent this works for the eukaryotes, I'll let you know in a year. If you guys get me back here, we'll start. So we're now running the Death Galaxy with eukaryotic cancer cells to see whether we can drive the evolution forward of the cancer cells to the metastatic transition or to evolve resistance to. We'll be doing that, but it's going to be a much more challenging problem, as you're saying, right? Because we will probably be seeing SNPs all over the place in non-coding regions, I suspect.
Apart from SNPs, can you build in something linked? For instance, the genes that you mentioned, gyrates, obviously linked to transporter, and obviously you have a gene map, so you can address your disorderness at a different level. If you have built in known, for instance, it is known that the gene map is linked. For instance, certain group of genes are more co-evolved or something like that.
Right, I'm trying to understand what you're saying. So we now know that all these genes co-evolve, right? That they're
gyrates, as you rightfully mentioned, is linked with the two other genes.
Yeah, that's right. Yeah, the gyrase is linked with the pumps, right? That's right. It's linked with the pumps. Now, I don't know whether that.
So many genes are also known to link.
Right. They're functionally linked together, right? They seem to be quite far apart, and nonetheless, they're functionally linked with one another. Right, that's right. Some of them are actually physically linked, right? So the,
So now your entropy can be...
It's very complex space, isn't it? Right. So again, whether it's a computational tool, just an analogy to understand the way the system works, that remains to be seen.
So now evolving not of genes, but of operons.
Yes.
Evolving of, as you also mentioned,
yeah, we see the MAR operon actually changing here. Right, yeah. What is surprising to me is the system came up with such a parsimonious solution to the antibiotic question so rapidly, and it keeps up with the same thing every time. So apparently there's a very unique solution that lies quite deep in free energy space, right? And it's able to get there really quickly. I find that very weird, and I don't understand it.
Is just about that. It's because every time we think about evolution in general, not in bacteria, we always say that the origin of evolution is diversity in any population. And in your experiments, for example, when you start with 100 bacteria, you don't have that diversity, and that's what amazes us, right? That you can find the bacteria that evolve even when there is no such diversity.
Right.
So I wonder if in those conditions, you are not promoting some kind of diversity in those bacteria, that there's some kind of mechanisms where they can, I don't know.
They must be generating diversity in order to search through this space, right? That has to be happening, right? That the search occurs through a opening up of diversity, and then they seem to close it back down again. So I think that the diversity is a function of the path along this space. We've actually done other I didn't show the data here. We tried chlorophenicol. You know chlorophenicol?
Yes.
It blocks ribosomes. It's a metabolic poison. It's not a genotoxic poison. So because that doesn't go directly after the genome and doesn't cause error-prone polymerases, you would think that if I try the same experiment with chlorophenicol, that the evolution to resistance to chlorophenicol will be slower than it is with the gyrates, because the diversity, the search over entropy space, is smaller. And that's true. When I can show you the data. It takes 30 hours in my device for my bacteria. They do evolve resistance to chlorophenicol, by the way. But it takes 30 hours where it took 3 hours with Cipro. So it is, in fact, a strong function of the transient diversity that the bacteria generates as it searches through entropy space to try to find a new minimum in free energy. Does that help at all a little bit?
Yeah,
Hope I answered that question
I think that
a little
helps.
bit.
Thank you.
Yeah.
Any more questions? Yes.
Okay, I have a question here. Because some bacteria could get some plasmid by accident, so how do your system to get with this problem?
I didn't understand it. Did you follow that? My hearing is terrible, as my wife knows. It's not you, it's me, by the way. Yeah.
I see some bacteria could get some extra from the outside environment plasmid by accident.
Plasmids.
Yeah, some bacteria.
The bacteria that in this original experiment don't have the fertility plasmid in them, so they basically don't. They cannot conjugate and transfer information around, right? So the only plasmid, and the GFP is actually, there's no plasmids on these bacteria that we're working with. But, I mean, plasmid transmission, that's what I was trying to say in the end, is certainly a way that evolution occurs, right? And we're going after that. But these original experiments, it's not plasmid because everything is on the chromosome of the E. coli. The first experiments I told you about, right?
Thank you.
So we just shut that off because it doesn't mean they're not chopping off the DNA and putting it on the solution and picking it up again. They can do that without plasmids. We know that's true also. But at least in these experiments, no plasmids. I think I can state that with confidence. But it's certainly a way that evolution can occur in the system. Don't get me wrong. We just didn't want to have that happen. Okay? Hope I answered that.
Yeah.
Microphone, because my hearing is awful. Oh, there we go.
Okay. I'm wondering how confident are you on those concepts that you mentioned? This Darwinian thermodynamics. Is it any good, do you think?
Sure, why not? It's based on such general things that can't possibly be wrong. It's non-testable. It's bullshit, in other words. Yeah, I think it's of such generality what I said there. It'd be hard to see how that could fail, right? What I told you, right? How would that? It'd be hard to see why what I said is not probably correct at some overview level. I think, but I mean, of course, everything gets screwed up if. I'm standing on the edge of a cliff. Because there's, you may know about the experiments of John Cairns and other people back in the 70s where they thought there was directed evolution going on. That when the organism sees something and makes mutations in a particular place and not elsewhere. It's not random. So my arguments taken out of classical statistics and physics are basically assuming that everything is random, right? If the system has strong non-random aspects to it, then probably what I'm saying is not so much wrong as irrelevant, right? So that's a possibility. And since I haven't actually tried to compute the 3-hour evolution time I see, it could be true that the system is making very specific mutations at a particular point. That's not part of physics, right? Physicists don't talk like that. And so that could be happening. So I guess it is testable in the sense that if what I told you, if I try to compute my entropy and find out that there's no way in hell it could have searched through that entropy space so rapidly, then there must be something wrong in my basic premises. So that's testable. Say again? Go ahead.
The thesis assumes that the bacteria is responding to the antibiotic.
Right.
But there doesn't have to be an antibiotic. The bacteria could be doing this on its own.
What does it mean? In the absence of antibiotic? Yeah. Well, that's Luria-Dilbrook, right? It says that, and it's sure that's, yeah. Sure. Yeah, right. Absolutely. Right. The bacteria generate mutations ab initio. That's that 10 to the minus 9 rate. They're making them all the time, but at a very low rate. It's all a question of, okay, I know that happens, but I saw evolution in a very specific way happen in 3 hours in 20 generations. If it was Luria-Dilbrook, just a thing just randomly making mutations at some known low rate, does that in any way, shape, or form predict the rate at which I saw evolution occur? If it's off by orders of magnitude, I would claim it's off by 3 orders of magnitude. Then it must be true that the mutations we saw were in response to the antibiotic. But that's not a radical statement because the SOS response is known to generate.
Without the antibiotic.
Say again?
If you run it just with the bacteria.
What kind of a test do you want me to do?
Your experiment.
What kind of a test?
So a control. Yeah, control.
What is it? Tell me the experiment, and I'm sure I've done it. What do you want me to do?
Just 1 or 3 hours, and we test how many resistance to Cipro.
What do you want me to do? I'm sure I've done it. I've always.
Then what you're asking, so
Finally, we get down to brass tacks. All right, tell me what. come up with something. I'll tell you the experiment I've done.
I have an idea.
I don't see why the bacteria would stop mutating on its own.
They don't.
Well,
Right, they absolutely don't.
it doesn't matter what the antibiotic.
Say again, now again.
So the control would be you run the experiment for 3 hours, and he probably want to know how many Cipro resistance.
You won't find any.
You won't find any. So that's the control.
If yeah. They will, but it's going to take you 30 days to see.
The rate.
It's all about the rate, right? It's all in the numbers. It's physics, right?
So that's the
Yeah, there's nothing wrong with what you're saying.
Another antibiotic, and you're going to have a different.
Right, I just told you that chlorophenicol, for instance, which is not genotoxic, takes 30 hours to generate for us to see the emergence of resistance because chlorophenicol.
What's bothering me, I don't see how
Yeah.
you can stop the bacteria no matter what you do.
Oh, I'm not stopping them. I'm turning them on, not turning them off. Right? The sign is that I'm upping the mutation rates. So there's two things going on. The mutation rates are going up because it's a genotoxic antibiotic. It ups the mutation rate by 2 to 3 orders of magnitude. The second thing that happens is I create a landscape where if you are a mutant, you can go to a place where you can compete and fix rapidly. So there's two things going on simultaneously. And I can run an antibiotic which is not genotoxic. So it's slower now because I'm now presumably running at your basal level of 10 to the minus 9. No argument that it's always there. But now once a mutation does happen, if you wait long enough, then it can rapidly fix because I have this landscape where it flows down. Right? So there's two things going on simultaneously. That's why I rate a free energy. Because free energy takes both those things into account. It's not heretical. It really isn't.
Last question on the back. You have anybody?
My question is, it could be this you find several mutations, right, from the Cipro resistant.
Yeah, we see 4, right.
But I understand this professor argument is whether this is due to the Cipro specific or it driven several type of mutation. One is general mutation, which will speed up further mutation, and other is some specific mutation that caused by Cipro. Could you modify your experiment to answer this question?
What do you want me to do?
Let's say in one of your experimental results, in the growing, the growing condition not favored for growing, but without any antibiotic.
Yeah.
The growth rate in the first non-selected bacteria is growing faster than the side with antibiotic. Could you could you grab the cells from this selected area? That means the first one is not favored for growing, and put back to the assay, put back to the chips.
Oh, you want me to re-put them back in?
Yes.
Yeah, we've done that, and now
you did it.
Yeah. Oh, yeah. Believe me, I have not told you all the experiments I have done to beat down the biology.
But what is the result?
And so they just immediately go out and grow. Right, so the mutant, I know what you were asking, but we take the evolved bacteria and re-inoculate them in the chip, then the time course of growth is completely different. Before they had to evolve, and then now they just grow everywhere on the chip.
Even in the Cipro.
Both places. It turns out the fitness of these evolved bacteria is only slightly depressed under normal conditions. Right, they grow almost as fast as a wild type, not quite. So they're capable, as you saw, of growing around the entire chip periphery. It's not like they only grow in Cipro. These guys can grow in Cipro, but they can grow quite happily, almost the same fitness in the absence of Cipro.
But then I cannot understand.
You don't want them to get loose.
This I cannot understand because this one is not Cipro selected cells, bacteria.
Right.
But it can grow same in the Cipro administrate area.
Right.
This not make sense, right?
Right. It doesn't come at a great cost of fitness to become Cipro resistant. It has found a very good solution which gives broadband resistance and doesn't cost it a lot in terms of fitness in a normal environment. Right? It's a very clever solution it's come up with. Am I answering your question at all? It doesn't create they're not specialists. The superbugs grow anywhere.
But the growth rate, you mean even the rate is the same, growing faster in everywhere?
Yeah, you're right. I'm not getting where you're going.
Maybe I rephrase my questions. You
have
to.
I don't know. Say in the ciprofloxacin, in the Cipro area, the bacteria grow much slower, right?
What's that again now?
The growth rate.
The growth rate of a bacteria in the Cipro.
The wild type.
The wild type.
They can't even. They can't even grow at all.
In other words, in your 3 hours of experiment, the first say,
nothing much is happening at all, right?
The bacteria will be just doing nothing.
Well, I wouldn't say nothing. They have to be doing something, right? They have to be in in other words, you have to create a place where there's what's called a MIC, the minimum inhibitory. You have to have some replication happening to evolve, right? So there are places where it's some. That's why that gradient. They're at a place where there's almost no Cipro and very high Cipro. There's growth occur. You have to have growth, but once the mutation happens, kaboom, then they just take off. If you run the chip with Cipro everywhere, you see nothing either, right? We're way above the MIC, right? You have to have the gradient. Believe me, we've tried everything. You can't beat me. Whatever you I will win. I've done every experiment.
Okay, so because of time limit, let's just close this session. For those who still have questions, please come to here and ask more questions. Finally, let's thank Professor Austin.
Thanks, Pinker.